Yeah, we are live on YouTube now.
Okay.
Yeah, if you want, you can switch off the camera for some time.
Okay, and I'm just thinking.
Yeah, you have all the controls with you.
Yes, now I see.
Yeah, yeah. Okay.
So good evening, all of you, and welcome to the final day of lecture by Dr. Nagma Parveen as part of the COVID week. But before I introduce today's speaker, myself and Professor Harish, we want to place vote of thanks to all the people who have supported to have this initiative on ground. So first of all, let me thank Professor Vinod Singh from chemistry. So he was the one who approached Professor Harish with the idea that we should have a wider talk within the IIT Kanpur community by experts who are working in this area. And we all look for various journals or news media article or WhatsApp things for understanding questions which are quite obvious related to COVID, whereas we have among ourselves such experts who are working day in and day out and know the real truth behind these things, or they are trying to get that. So it was wonderful to have all the speakers, starting from Dr. Divyendu, Dr. Amitabh, Dr. Vishal, Dr. Manind, and Dr. Nagma, to be so generous and ready to share their insight and the research which is still upcoming. And they explained things in quite detail and were very generous to answer a lot of queries which were raised after the seminars to them. And hopefully today also we'll have a lot of these questions to be answered. Similarly, there are support from Professor Abhay Karandikar and Professor S. Ganesh in organizing this talk. Apart from that, I would like to thank Professor Nishant Nair and Professor Satyaki for helping out with the logistic and webinar issues, and Mr. Sanjay from MediaCell for helping us with the YouTube platform, Mrs. Chitrilekha from R&D for the poster and support with the slides, and anyone else that I might have missed, and of course a wonderful audience who have posed very nicely all the questions for the benefit of both wider audience and people who watch this on the YouTube. So some of these are available on the YouTube and people can go and look at it. So thank you very much. Now it's time to introduce today's speaker. So we have Dr. Nagma Parveen. She joined last year as faculty in chemistry department at IIT Kanpur. She has her BSc from University of Calcutta in chemistry and MSc from IIT Madras. She has done her PhD from University of Münster, Germany, and 2 postdoc, one from Chalmers University, Sweden, and one from University of Leuven, Belgium. So she has a lot of experience, and let's hear from her. And any questions that you have, please put it on the question answer box or raise your hand or put it on the chat box. I'll try to unmute yourself one by one, and then you can ask after the seminar all these questions to Dr. Parveen. So please go ahead.
Thanks for the introduction, Professor Gupta. I will just start. So this is the outline of the talk. So I think I did not write that I will talk about therapeutics also. So I will also tell you about the therapeutics of the COVID-19. Okay, so I will start with the immune system. So when our immune system is quite actually strong. So whenever we are attacked by any pathogen, bacteria, virus, fungus, yeast, so our immune system actually works very well to either engulf them or to avoid them somehow. So the system which works together is immune system, and one part of the immune system is called innate immune system, and skin, mucosal layer, acidic pH in our stomach, all are part of this innate immune system, and some specialized cells that are called natural killer cells. Okay, so like lymphocytes, macrophages, neutrophils. So here is a video. So if I start from the beginning, you see these are macrophages, and when yeast, which are pathogens, which comes in, these cells can engulf them and send them to their lysozyme, where they are broken up, they are like a chopped up, and then in this way they kind of eats up, if I say in a very layman language, these pathogens. But this innate immune system's working is very generic, so it can engulf any pathogen or any outer body part, like which is not part of our body, or which is not part of our system, that can be engulfed. And how this works, this unspecific engulfing works via these toll-like receptors. So in our plasma membrane and also inside our cells, we have these receptors, this Y-shaped receptors, and they can sense some lipopeptides or liposaccharide, or you see these flagella, these flagellas on a bacteria, and then they can engulf them, and also they send a series of signal, as you can see here, and they send the signal to our DNA to produce some special molecules, and these are called cytokines or chemokines, and they are actually nothing but inflammatory markers. So once we are invaded by some of these pathogens, these receptors job is to signal our body, our DNA, to produce these inflammatory markers so that our immune system become more active. So in this way they work. So this is a very simplified version I'm telling. So that is innate immune system. What about another part of the immune system, that is adaptive immune system? So by now you all know about this antibody. So this antibody is a protein which is like this Y-shaped protein, and this has the constant part. So it's written as C, a light chain and heavy chain, and this, the top of this antibody, is the variable part, so light chain and heavy chain. And this part of this antibody is very important, this variable part. Here there are specific amino acid sequence through which it can bind to antigen. For example, for SARS-CoV-2, we have the spike protein. So the spike protein is this one, let's say the amino acid sequence of the spike protein in here, and the antibody binds to like a lock and key model. It's kind of fit together, and each antibody is very specific to particular antigen. And this antibody's job is to neutralize any pathogen enters in our body. And who makes this antibody? We have a B cell, and that B cell is a part of adaptive immune system that actually neutralizes these pathogens, and they are produced, they are generally on top of our B cells, like a flagging out. So our body produces this B cell, and these are called naive B cell, and the naive B cell then differentiate into two parts, the plasma B cell, and plasma B cell's job is to release this antibody in our plasma. So you see they are called IgA, and this is the first type of antibody when we produce. And then another very important cell is produced, and that is called memory B cells, and these we need because this, as the name says, memory B cell. So once these cells are produced, they stay with us for 50-60 years, and the antibodies stay on its surface, and these antibodies are IgM and IgG, and this is their structure. So eventually we want to produce IgM and IgG, preferentially IgG, because it can be transferred from mothers to their newborn babies via placenta. Well, this is B cells' job, but this innate and adaptive immune system, they do not work separately, they work together. And I did not mention about a very important cell, which is the dendritic cell. So I will try to give an overall look how our immune system works. For example, when we get infected by a virus, or when we get a vaccine dose, for example, then we have a special type of cell. These are called dendritic cell, and their name is also antigen presenting cells. So what they do, they are part of our innate immune system. So they take this virus, they engulf the virus, chop them up, and then they present some part of this virus on their surface. So that's why it's called antigen presenting cells. Okay. And then we have some specialized cells that are called T cells, CD4 T cells. They are part of our adaptive immune system. They come up, they bind together, and once they bind together, they send some signal via cytokines, and that tells our B cell which antibody to produce. So I told you that B cells produce antibody, but this is the mechanism how they make particular type of antibody against particular pathogen. So this is very specific pathway. This is one pathway. And then, as usual, as I said, that then B cell produce IgM, IgG, and IgA. Another pathway is this antibody presenting cells are detected by CD8 T cells. This is another. So 8 and here is 4. Okay. So these CD8 T cells are very special. They again go and bind to these antigen presenting cells, and then they present a small part of the virus, not the spike protein, for example, but a small peptide unit, very small part of the virus. So you can imagine, like, it is not only limited to large proteins of the virus, but also small peptides of the virus can be detected. So then it presents the on its surface the peptide, and now these CD8 T cells can divide, differentiate, and form memory T cells, and the peptide of this virus stay with us for at least 10 years or more. And there are also killing infected cells. So this tooth cells. So at the end, what we want from our immune system, that we produce enough memory T cells, enough memory B cells, and good amount of antibody, and preferentially IgG. So generally, when we are constantly evaded by some pathogens, bacteria, viruses, and our body actually works very well to do this job and keep these memories and make these antibodies, and we produce them, these antibodies, in our plasma, and that is called convalescent plasma. However, in case of SARS-CoV-2, it's becoming very much difficult, and I will come to that. But how? Let's see where actually this SARS-CoV-2 attacks. So SARS-CoV-2 actually infects our upper respiratory tract, that is this part, and then also lower respiratory tract. So in the upper respiratory tract, it attacks these cells, these olfactory cells. That's why we lose many people, not everyone, lose their sense of smell and taste, and also it relates to the neuronal cases, which is more and more becoming clear. And in the lower respiratory tract, it infects in bronchi, this kind of ciliated cell, and most importantly in this alveolar. So the end of our end of our lungs, where these alveolar sacs are there's this special cell. It's called alveolar epithelial cell. So this virus goes there, replicate there, exhaust the cells, and the cells start to die. That is how our lungs start to dysfunction. And when actually, and how the replication happens. So for example, if a person get infected and get a symptom of fever or something like that, that is we call 0th day, and actually the viral infection has started 4 or 5 days before. So the virus has entered in our body and started to replicate 4 or 5 days before the symptoms come, and then it peaks about on the 2nd or 3rd day from the symptoms, and then the replication goes down because our antibody starts to produce. So our immune system starts to fight against the virus, and virus replication goes down within 15 days. So this is a normal thing. And I also must say that this data is from viral replication on this part, and we don't know what happens in the lungs, because the swab test is generally done from this upper respiratory tract and not the lower respiratory tract. So what are the symptoms so far? So there are a majority of the people who are actually asymptomatic, as we know, because they get infected, they have all this replication going on. However, they don't get any fever or any cough or any symptom. Mild patients, they have some fever, fatigue, and sore throat and other things, but they recover within this 15 days from symptom. And moderate patient develop some pneumonia. So the infection goes in the lungs, and our body is not capable to really fight in time. And severe cases, it goes bad. The pneumonia develops further, and in critical cases, the respiratory system start to fail. However, as it is known that 80% of the cases we see asymptomatic and mild cases, and we can recover at home, but 15% of the cases there is a moderate to severe disease, and 5% of cases reaches to the critical ill, and some of them unfortunately succumb to death. So actually the problem with SARS-CoV-2 is that we still don't know how it is causing the disease. The main reason is because there is no proper animal model for this virus. This virus is causing severe disease to, for example, hamster, but not to other animals. So it is becoming very difficult for scientists to know how it attacks our lungs. So, but people have studied other coronaviruses like SARS and MERS and influenza. So from that, they can have an idea what is the possible mechanism at cellular level. So what happens? So this is our healthy lung, and at this end of this lung we have this alveolar sac. Yeah? So the alveolar sac is nothing but air bag, and around the air bags we have this blood vessel, and we have this epithelial cell which I said about earlier, and these cells are important to transport this oxygen carbon dioxide, to diffuse the oxygen and carbon dioxide from the alveolar sac to this blood vessel, which is very important, right, for functioning of lung. So generally what happens, naturally if we are healthy, these epithelial cells are forming and dying, and it's a very regular manner, 50-50. However, when we get infected by these pathogens, then the death of this alveolar cell is more compared to how much they are forming, and that then our body has to do something, right? So there is a second plan for the body. So they start to make this progenitor cells which transfer into epithelial cells. Okay, so there is some backup plan, and these epithelial cells then can replenish whichever this 10% lacking. However, if the viral replication goes very high and there is more death to these cells, then there is no enough time, and there is a different type of progenitor cells which starts to form, which is not like these alveolar cells. You see, it's quite different, and they kind of form this barrier, and they kind of clog this alveolar epithelial, this alveolar sac, and there is no exchange between oxygen and carbon dioxide anymore possible, and then how the lung starts to become bad. And this is also why we are getting hypoxia. This is SpO2 level is going down in the moderate to severe COVID patients. And then what we need is mechanical ventilation or external oxygen supply. So this is one type of pathogenesis. Another very highly prevalent pathogenesis in COVID patient is formation of pneumonia. So this side, the left side, is the healthy lung. So you see this alveolar sac and the blood vessel, and they are separated. However, when the infection really is high, then this alveolar epithelial cell starts to die, and then this barrier between them breaks down, and the fluid from this blood vessel and other areas enter into our alveolar sac. Okay, and then starts to damage our alveolar sacs in our lungs. So this is actually a autopsy, a sample from a COVID patient who succumbed to death, and it was some area of the lungs where this pneumonia has not happened. And you see this is the alveolar sac area, and this around it the blood vessel going on, and different cells are there. And severe cases of pneumonia, you start to see that this difference is gone. So all fluid, all alveolar sac is full of fluid, and this is how pneumonia is detected in a biopsy or autopsy samples. So this is how it's causing severe disease. And then the third pathogenesis of COVID is lymphonia. So what is lymphonia? So I told you in the beginning that when we are attacked by any pathogen, our innate or our adaptive immune system works against them and then produce these cytokines, right? And this is important to signal. However, if these cytokines are forming too much in our body, if there is a cytokine storm in our body, then it's becoming like an autoimmune disease. It's becoming like our body working against us, and that is what also is seen in a COVID patient from going from moderate to critical. What they are observing in the clinical studies, that these different markers, the different cytokine markers, interleukin and chemokine, their concentration is highly increasing, or their expression is highly increasing. in people who are diseased and who are in a critical conditions, and that's why almost in last year August they have come up that it is better to treat severe patient not for antivirals but drugs which actually suppress these markers, or which suppresses our inflammation. And also what is becoming more clear that in COVID patient, their memory T cells are actually lacking, or the memory T cell against this virus is going down. So this is the healthy patient, and then the non-ICU and ICU cells, you see the T cells counts are going down, and also depending on the disease severity. So the hypothesis is the naive T cell, when it is a mild disease, it's differentiating properly and making the memory T cells. However, if the disease severe and the cytokine storm is too high, then the memory T cells are few producing, and it's exhausted. The T cells production is exhausted. So together, what happens in the pre-symptomatic or asymptomatic case, mostly this virus is attacking our upper respiratory tract. In the symptomatic and early phase of infection, it is already starting to infect our lower respiratory tract, and that's why we are developing all the symptoms and a very low level of pneumonia. However, in the moderate to severe, we are having a high level of pneumonia and the cytokine storm, which further leads to blood clot. So all these three together is causing this COVID-19 disease. And it is increasingly clear that it is highly related with male sex, because they are suffering from severe lymphonia, whereas women have a higher CD4 and CD8 T cell activation. It is also more prevalent in obesity because they have altered T cell differentiation and also a difference in interferon level. And also it is related to age, particularly older age. I don't know whether there is information about the kids or newborn, because as I said, our memory B cell lasts with us for 50-60 years. So as we grow older, the memory B cell deteriorates and our immune system deteriorates. So it's also attacking more to the older people. And also there is a European study, particularly in Spain, and where they saw that A blood group people are at higher risk compared to O blood group. And most importantly, it is associated with comorbid diseases like people with diabetes, heart disease. And I was trying to look at how these are related. So it was a bit difficult to find out. But what I found out, that some people are saying that our pancreas, so people with diabetes, their pancreas do not function properly, particularly the beta cells of the pancreas. And this beta cells actually expresses ACE2 receptor. This is the receptor for the virus. So virus binds to this receptor to enter into cells and then start to replicate there. And this pancreas has a lot of ACE2 cells. So it is possible that the virus is also attacking our pancreas, infecting our pancreas and causing hyperglycemia or ketoacidosis. Heart, of course, because this ACE2 receptor is a very important receptor for our body, because this is our body part. They have a very important function, and the important function of this receptor is to control the blood pressure when our heart pumping the blood. So the blood pressure is important to control, and that is this enzyme job to transfer this in a cyclic manner. And then it is possible that the virus is directly infecting the heart. And also we already know that heart and lungs functioning are very synchronous, right? Because the oxygen loaded blood is going to here. So they are very together working, and hence if the lungs start to get infected and not properly function, the heart's condition will go bad. So together, these comorbid condition is increasing, is making it more severe cases for COVID-19 patients. So how should we treat it? So the best, or if someone think of, the ideal way will be to target the virus, right? And how should we target the virus? Let's target how the virus is entering in our cells. So that is one way, and that's how this hydroxychloroquine came, because it can interrupt its endocytosis process. However, so this is just a chart of COVID patient at standard care, and you see 130 people reached to mortality out of 1,000. So these are number. And when they were treated with hydroxychloroquine, the mortality actually went up, so +10, and mechanical ventilation went up. But when it is treated with a combination of azithromycin, it went down. But this gray data means there is not enough size of the sample of this clinical data, so we cannot really trust this data. So that's why hydroxychloroquine was dropped. Then another thing which we can target for the virus. So the virus has different enzymes in it, so they are special to the virus, so that virus can replicate, and one of them is protease. And if we can target that, that will also help, because then the virus cannot replicate anymore. And one of such, actually two of such drug is lopinavir and ritonavir, and they were actually they're used drug for against the hepatitis C virus. However, these drugs which works against this viral protease also did not show good result in terms of mortality, mechanical ventilation going down, or and also introduce adverse effects, and also the sample size was low. And then third possibility is to target the virus polymerase. This is a very important enzyme of the virus, and particularly for a RNA virus. So I will not go into details. And this allows the virus to grow its RNA chain. Okay, so if we target this, if our drugs target this enzyme, then we can stop virus replication. So there is many drugs, RNA dependent drugs, and one of them is favipiravir and remdesivir, and they are used for Ebola, influenza, and HCV. So favipiravir shows a lowered mortality, mechanical ventilation, although some adverse effect, but the sample size was small. The good thing is the remdesivir showed a good result in terms of lowering mortality and mechanical ventilation. And this green shows also it has quite a size higher amount of clinical trials. And so I will just list the drugs which are used for the treatment, or at least the data I got so far from the published paper. So the standard care for a COVID patient is giving antipyretic, that is paracetamol, mainly paracetamol and not ibuprofen actually, and reducive, that is palmocular and Benadryl, so generally for throat sore what we use, antibiotics like doxycycline, and multivitamins. So if someone gets a mild disease, corticosteroid, which is also commercially called budesonide, is used. This is an asthma drug in this dose. And the phase clinical trial was done with 146 people. So you can see here divided into groups, control and budesonide. And you see if the patient of mild disease are given budesonide early on, their recovery is earlier and also in a higher percent. And also it is only one clinical data, but many clinical such data has been done, and it shows a very green. So green means there are multiple data, so it's a strong data. Then for moderate disease, remdesivir, which is an antiviral that is used in this dose, and the clinical trials is quite a big size, 1,062 patients divided in control and patient group. And you see the remdesivir, if given to moderate patient early on, then the recovery chances are higher and also earlier, slightly, not really earlier, but slightly higher. And the clinical trial data size is also quite good. And then for moderate to severe disease, and this is most difficult, and here what is shown a good result is glucocorticoid, dexamethasone, with this dose, and this size of the clinical trial is also quite big. And what they found out that in this clinical trial, that among about 22.9% people in this in the trial group was succumbed to death, whereas in the control group with standard care, the mortality rate was slightly higher. So here is the plot. So you see the mortality rate is higher in usual care, where with dexamethasone, if given early on, it goes down. So I just made a list because there are also few other drugs like ivermectin for mild diseases, then the tocilizumab. This is an antibody against interleukin. So interleukin, you remember this is the chemokine, which is this chemokine storm we get. So against them. And in case of this antibody against IL-6, this drug actually do not improve the survival, but it reduce the mechanical ventilation time. So in general, if you look into this list, what we found out that in this list, majority of them are actually steroids or anti-inflammatory drugs and not really antiviral. Only one antiviral sits on this list. So this tells us that we are really not targeting the viral or viral replication, but we are targeting all other things in our body to treat COVID-19. And if you are interested, you can go this paper, which has actually collected 189 publications data and made this really nice chart. And if you're interested, you can look at different combination of drug has been used with different certainty and different type of analysis they have done, and they have just collected the data, all the clinical trials data, and made this chart. I also want to tell now a little bit about this other diseases which we are seeing now, that is this mucormycosis. So COVID patients, severe COVID patients are also getting this fungal disease. So this is the fungus which actually infects the sinus and the brain, and also another type which infects the lungs. This fungal infection is seen in severe cases, severe COVID patient, and this is becoming surfacing in India, and there are some reports which was in China but not in Europe and US. And interestingly, what I found that in PGIMIR they published the mucormycosis was associated with COVID-19 cases early in January. So they must have observed it last year. And they have told about that the use of this glucocorticoid, dexamethasone, is possibly suppressing the inflammation in our body, and then other pathogens are popping up, and they can take the advantage of lower immunity of our body and infect. That's a problem of this kind of steroidal drug, that they are not really targeting our virus, the viral replication, but our immune system, and that's why other pathogens can take up and cause severe diseases. But the good thing is mucormycosis can be treated. It has a drug, and this is the drug amphotericin C, which is generally formed in liposomes and then given, which breaks down which makes pore, actually takes out the sterol of the fungus and kills the fungus. So it targets against the fungus. So if it is diagnosed early on, this can be treated. But I want to tell you a little bit about new class of treatment, and which maybe we should a little bit more focus on, and that is antibody treatment. So this is the spike protein, part of the spike protein of the coronavirus, and this is the antibody which is produced in our body when we are infected. And many antibodies, I'm showing just two, so you see how they bind. So people are now thinking to make this antibody, or not thinking, they already have done in a humanized mice, and then collect this antibody, and then in the laboratory they could see that this combination of antibody can really neutralize SARS-CoV-2. That means if they can be administered in patients, they can treat, because now this is targeting directly to the virus and neutralizing the virus. So actually Roche, the company, has already this two drug combination, this two antibody drug combination in phase 2 clinical trials. And another company, Eli Lilly, has a very similar type of antibody which they made, which is, these are the name, and these are already is used in emergency use in USA. And these phase 3 clinical trial data show that 3%, so if the people with mild COVID disease are treated with this drug combination, antibody drug combination, they have 3% of chances to admit hospitalization in compared to control group, where 10% of the people end up in hospitalization. Okay, so now I will move a little bit about the importance of vaccine and how to design vaccine. So this paper is very nice, and if you are interested, please go and have a look. Here's the DOE of this paper. So here, what they did, they tracked 188 COVID patients with mild to severe disease who are like recovered. And they tracked them. They did the kinetic study for 8 months. And what they did, they looked at the how much memory B cells they produced against the antibody against the spike protein, different parts of the spike protein, including receptor binding domain, then nucleocapsid. And then also they looked how much memory T cells they produce, CD4 T cells, CD8 T cells. And when they looked in the first month, you see this black group, they have all. They have the antibody, they have the B cells, they have the CD4 T cells, CD8 T cells, and IgA. And you see after 6 months, the pink group is rising, and the pink group lacks the CD8 T cells. So they lack this 8 minus. So this is what is the overall conclusion. But I will just, okay, I also wanted to say that we always talk about antibody against the spike protein. And that's true, that is we produce, of course, but we also produce antibody against nucleocapsid, okay? So we have many types of, nucleocapsid is also part of this virus. And when we do this rapid antigen test, this nucleocapsid is used actually to detect whether we produce the antibody against this nucleocapsid. So this antibody, we also enough amount we produce when we get infected. So in this paper, what they followed, as I said, they did kinetic study. So what they found out, the antibody against the spike protein of the coronavirus was slightly decaying over 6 months of time with a T half-life time about 140 days. Whereas for nucleocapsid IgG, the antibody against nucleocapsid was having a lower lifetime, about 68 days. And the memory B cells was intact for 6 months. So that's good that once if we are infected, it stays there for at least 6 months. However, they find that the CD4 T cells and CD4 T cells were 8 cells, both T cells were declining. So with the apparent T half lifetime about 100 days. And this is bad because if we compare, for example, if a person is infected by polio disease, polio virus, and they produce T cells which last for at least 10 years. So this lowering of T cells is not good, and possibly, I don't know whether this is the reason, but it is possibly indicating why we are getting reinfected, because the T cells decline. Okay, so now I think you got an idea that what should we look for when we make a vaccine. We should look for antibody, we should look for the memory B cells and memory T cells, and of course their kinetics over time, how they are changing, right? So this is a SARS-CoV-2 cartoon, and this is, I think you have seen it multiple times, and this is a spike protein. So when we make a virus vaccine candidate, there are many types. So the old school type is just to take exact same virus and inactivate the viral RNA, and that can be done chemically. So then this vaccine, this is a vaccine candidate. It cannot replicate, but it's there. It has all the proteins, and it has the immunogenic property. So if it is administered in our body, then our immune system will work against the spike, the nucleocapsid, all the other viral protein, because it is something outside of our body, right? This is one candidate. Another candidate is not completely inactivate, but partially, and that is called live attenuated vaccine, very similar. And then another possibility is to just take the viral protein, in this case, the spike protein, or take a small part of the spike protein, like a recombinant receptor binding domain of the spike protein. Another is virus-like particles. So just take a hollow virus. So there is no RNA inside at all. Another, which is a new class of vaccine, and that because of all this COVID disease, we got this first time, this kind of vaccine, and that is a vector-based vaccine. So in this vector, so you take external virus, another virus, in this case, it was adenovirus, which acts as a vector, and in the DNA of this virus, you put cut and paste the gene of the spike protein. just parts. So that once this virus goes in our body, then it goes in our in our cytoplasm, and then release this DNA, the spike gene, and the spike protein is formed, and then our immune system make antibody against the spike protein. So this is the idea. So one, we can do the replication incompetent vector. That means this viral vector, the DNA of the viral vector is suppressed, so it cannot replicate. So it once goes there, once it can deliver the spike gene, only once, and cannot keep on dividing. Or we can also make replication competent vector, which can replicate and keep on making the spike gene in our body. Another is take another type of virus and put a spike protein on the virus surface, like a chimeric virus kind of thing. Another is instead of putting a vector, directly take the DNA and put the spike gene on the DNA and deliver it. No vector around it. And another very novel class of vaccine, and that is mRNA vaccine. So this is RNA, which is a single-stranded nucleobase chain, and cut paste the spike gene in this RNA, and to protect the RNA, because RNA is it really gets broken easily, cleaved by enzymes. So you put in a liposome, in a lipid vesicle. So for SARS-CoV-2, these three has been actually working very well, and these three has been shown good result, these three types. So inactivated type, then vector type, and mRNA type. So you can see all three are replication incompetent. So once they go, they are just once go there and then deliver the protein, viral protein or viral DNA or viral RNA. So that's why it needs a prime boost. So once it is gone there, then our immune system works against them, and then just to make sure that it works for longer term, we give another boost. So we get this prime boost vaccine. Yeah, and together with this vaccine, we also get additional molecules, which is called adjuvant. So I will not get into details. Adjuvant are some additional molecules which are not directly immunogenic, but they helps to trigger the immune system, such as aluminum hydroxide, certain types of lipids, and certain type of small molecules like imidazoquinone. So generally, when vaccines are made, it takes a very long time. So you can see it takes about 15 years or so, because it goes this linear process in the laboratory work, then a mouse or other type of non-human work, and then it goes to the different clinical trials one by one. But in case of SARS-CoV-2, it was very amplified, as already we had an idea about this virus from the SARS-CoV and MERS-CoV epidemics, and also the clinical trials were done not in one by one, but all together parallelly it was going on. So it took only 1.5 years. And what generally is done in a vaccine clinical trial? So in a vaccine clinical trial, the trial is done in a random manner and double-blinded, so that who is giving the vaccine do not know what they are giving, and also who is receiving and they do not know. Then we take a non-COVID enroll. So the person who is getting the vaccine should not have COVID throughout this trial. Or they have to be dropped if they get the COVID. And the placebo group gets either buffer or adjuvant solution. And the number, now this is a little bit has been critical. So the number goes from 300 to 50,000. So if it is in hundreds, then it's a phase 1 clinical trial. If it is in thousands, then it is phase 2 clinical trial. If it is in 10,000s, then it's a phase 3 clinical trial. And of course, they have to take what the age group, sex, ethnicity, body mass index, all these parameters into account. And after these clinical trials are done, or during it is done, what analysis is done, that whether the people who receive the vaccine is developing any disease or lung infection. And of course, most important, that they have to look how much antibody level is rising, how much immune activation is happening, like how much memory B cells and T cells are producing, and what kind of adverse effects are happening in a 28 days interval. The thing is, how many types of vaccines are available? So I started with this Bharat Biotech vaccine. So this Bharat Biotech vaccine name is this BBV152, and it is this old school type inactivated virus vaccine. And it is in this dose, and the prime boost interval is 4 weeks, and the efficacy we still don't know, as the critical phase 3 data is not out. And this virus was isolated by NIV and ICMR. It is inactivated with propiolactin, and it has an adjuvant, algyl or alum adjuvant. And the good thing is the interim data show that the people who got the vaccination in their phase 2 data, and then they collected their plasma and the antibody, and they showed that the people who are vaccinated, their plasma can neutralize SARS-CoV-2. So in different time intervals. So they looked at 40 days interval, 56 days interval to the vaccination. So that's good thing. And also the people got vaccinated, they have a higher amount of T cells production compared to control and pre-vaccination. And also recently they published that the antibody produced in the vaccinated people in the plasma, they can neutralize different strains of the virus. Like this is the Indian strain and this is the UK strain. So that is also good. So it can act against different viruses. So it can stop reinfection. Another type of vaccine, which is also interesting in Indian context, because we are getting this vaccine, AstraZeneca vaccine, and this is the name, and it is this vector-based vaccine. And this vector is adenovirus vector of chimpanzee, and it was isolated from chimpanzee Y25. So you have to know that this is a virus, right? So this can also cause immunity in our body, and if we already have antibody against this chimp adenovirus, then it can never reach to our target cell and deliver the spike gene, right? So that's, they have already tested. They saw that UK population have 0% antibody against the viral vector, and Gambia population have about 9%. And AstraZeneca also found out, so they did like 2 big phase 3 clinical trials. So before the prime boost time was less than 6 weeks. And they showed if the prime boost, the interval, if they increase more than 12 weeks, then the antibody titer was increasing. The neutralization capacity of this antibody was increasing, as you see here. But this is only true for age less than 55, not above, because above age 55, it was not increasing. And it also has a good CD4, CD8 T response. And there was only two fatality in their trials. And I also want to say that the efficacy, as you see, it is 80% efficacy. So this does not mean that if 100 people are vaccinated, 80 people are safe and 20 not. No, the definition is this. The definition is 1 minus attack rate with vaccine divided by attack rate with placebo. And if you calculate, so you put the 0.8 this side, it will be, so let's say if you take 100 people and do not vaccinate, and let's say 20 people get moderate to severe disease, and if you take another 100 people and vaccinate, then only 4 people can get a moderate to severe disease. So it drastically lowers down this 80% efficacy means. Yeah, and the which is surfacing also recently now, that this AstraZeneca vaccine is not that good in neutralizing the UK variant, the B117, and that is true. It neutralizes the new type of, so if we are getting this AstraZeneca vaccine, we produce the antibody neutralizes the UK variant virus, but with a less efficiency. However, it protects us, just it's a little bit less, that's all. So I just tried to list all possible vaccines. So actually the first vaccine which was started was Sinovac biotech company in China, CoronaVac. And these are the details, and I don't know about its status anymore because I did not get much information on their phase 3 clinical data. And there are many other, and they also have this inactivated virus as a vaccine. They also have different companies which are making different type of vaccines. I did not look into the details. Then the Bharat Biotech, I already explained. Then AstraZeneca, which is vector based. Then comes the Gamaleya Center, that is the Russian vaccine, Sputnik 5, okay? And Sputnik 5 is also same as or similar as AstraZeneca vaccine, only the vector is not chimp vector, it's a human vector, and 2 types of human vector. One is Sero26 and Sero5. Sero26 is given in the first dose and Sero5 is given in the second dose. Then Johnson & Johnson or Janssen, and that is also ambitious. They want to make single dose. And they also have similar as the Gamaleya Center's vaccine, and they are phase 3. I think it is already out. I did not look. But okay, I want to say the Sputnik 5 also has a very high efficacy, 90%. That means only out of 1% may develop severe to severe disease, moderate to severe disease. Then another class of vaccine, which is maybe not coming to India so soon, that is Moderna's vaccine, which is mRNA vaccine, and it has a very high efficacy, 94%. Then Pfizer BioNTech vaccine, that is also mRNA based vaccine, and that is also 95% efficacy. Then there is another company called Novavax. They are using the viral spike protein directly as a vaccine, and I think their phase 3 is also out. I did not get the time to look into it. Then Zydus Cadila, they are making this DNA vector based vaccine. This Prema's Biotech, they are making this virus like particle based vaccine. So there are many vaccine candidates are coming in. So in just on a lighter note, I thought I can give you also a list of this company, and to just get a background with these companies are from where. For example, Moderna was established in USA, and a chemistry professor, Robert Langer, was involved in this company. Then AstraZeneca is driven by Oxford Vaccine Group, by Pollard and Gilbert. And so just, I don't want to go through these, but what I mean, so you see many companies like Prema's Biotech and Zydus Cadila, they are making new type of vaccine in India, and many companies are producing vaccines in India. So they are not innovating, but they are producing the vaccine. So India has a major role in the vaccine program. So now come the final part, that is this mutation. Okay, so we are so much hearing about the SARS-CoV-2 mutation. So I just want to give a little bit of idea what is this mutation. So when this SARS-CoV-2 came from China in January early, we had this virus D614. It looks like this, and D is aspartic acid. So in the 600th position, 614th position of the amino acid sequence, there was aspartic acid. And over time, it just mutated and became glycine, D2G, only one position. And all over the world, now we have the G variant. We don't have D anymore. So you see how the demography has changed. And this G variant is highly transmissive. So the number of outpatient has increased, inpatient has increased, and ICU number is also increased. And the mechanism why this G variant is highly virulent is not because, so if you see the where the mutation has happened, it's not on the head, this receptor binding domain of the virus, which everyone is talking, the head of the spike protein. No, it is somewhere in this conserved in this area, which actually allows the virus to fuse better or internalize better. So actually, that's why you see that how small change in the virus can change so much in their transmission. So it is important to know the sequence of the viruses and to track them over time. So the repository was created in GISAID. So please have a look. It's very, very nice repository, and you can see how much UK is contributing to this repository, a huge contribution. And in India, we are contributing by these organizations. So mutations, what happens that the one virus replicates, and generally these RNA viruses, they are very bad in replication because they don't have proofreading machinery. So when they from the parent to kids or progeny virus, they are not identical. There is always some small change, and then some of them become somehow more pathogenic. They can survive better, they can infect better, and they become dominant, and that's how a mutation comes in, and then become a new strain. So the variant of concern in this website listed is, of course, D614G, which is now everywhere. So it is no more of topic to talk. Then the UK variant, then the Brazil variant, and South African variant, California, there are many. And the Indian variant started with this B1617, but now it is established that B16172 is the variation of concern. And I want to just give you a look how less we are sequencing in India. So this is just yesterday's data. So this is UK variant B117, and United Kingdom has found 200,000 of them, and India has 748 of them. And if you compare, the Indian variant also is similar. The number of Indian variant and UK variant in India is very similar. And we are seeing this number because in India we are just not sequencing enough, because the sequences are coming mainly from West Bengal, Gujarat and Maharashtra, and very little sequencing is done. So we don't know which one is prevalent in India. Is this one or the UK one? So why also this UK variant and others are important, just to give a little bit of idea. So this is the UK variant, and it has many mutations, but why people got more talking point, because the receptor binding domain of this spike protein of this virus, there was a small change here. So you see here, this red one was changed. So people started to think, okay, if it is changing, then how the virus infect will change, and that will create a change in their transmission. The Brazil variant, in addition to that, they had this part was changed here, and also same for the South African variant. This part was changed, and this part is important because antibody binds near there. So if these viruses come up and transmits, then the antibody which we have produced, they may not kind of neutralize these viruses. So that's why it became concern. And I did not get much data on the Indian variant, but a Cambridge group and Indian group, they have now solved this structure of this variant. And they found also the variation or the mutation comes in here, which is very close to the antibody binding part. And also there is, which is not shown, is P681 position, which is also important for the fusion of the virus. But okay, but what they find out that this variant is not actually doing so much cellular entry compared to the Wuhan, the first variant, but it is causing cell-cell fusion. So possibly that is the reason that this variant can be of concern or can cause more transmission. So it is still very, very less information we know about. So that's why, because these variants are coming now, these all vaccine companies are really pushing to know whether their vaccine works against all these variants. And I think Pfizer BioNTech are leading in this regard, because they have done many clinical trials, and now they have shown that their vaccine, so these are their data, vaccine works against UK variant, and then this one is the Brazil variant, the neutralized, but the Washington, sorry, this is Washington, yeah, this is B315. This is the South African variant, but the neutralization of the South African variant is a little bit less. Bharat Biotech has also gave interim data that it neutralized the UK and also Indian variant. And AstraZeneca, unfortunately, their neutralization is a bit lower for UK and also for South African variant. However, the AstraZeneca has quite high efficacy in the vaccination. So together, I just want to tell that all these three types of vaccine which are available, they have quite a high efficacy. And now UK has released a very huge data of their vaccinated people. So you see how large is the size of the vaccinated people in the first and second dose of the Pfizer vaccine and AstraZeneca vaccine. And they released the data, and they showed that people who got vaccinated, their chances of infection, reinfection or after vaccination is lowered by 60 to 70%. So yeah, so if you get a vaccine, that's better, because the chances of getting infection and getting the disease is lowers drastically after vaccination. However, we also have to also careful, because this is a virus can be similar to the influenza virus, we can say, and influenza virus has a drug against it. It has a vaccine, every year vaccine, and still so many people die out of influenza, and every year we need influenza shot. So we should not be complacent, and possibly we need COVID vaccine every year, possibly if they because of their mutation or because of many other reasons, it could become like a flu, like an influenza vaccination. And also I want to stress that it is not only should rely only on vaccination, but for example, I put the HIV data. For example, the HIV, when the pandemic started, So after the pandemic started, people started developing drugs and antiretroviral drugs. And as soon as the antiretroviral ART therapy started, you see people living with HIV has increased. So they were not, they were dying, the people with HIV was dying, but as soon as the therapy starts and kicked off, then the death rate has really decreased and people can live a decent life with also HIV infection. So that's what I mean, that we should develop antiviral therapy for COVID, and of course parallel to the vaccination, and of course improve our medical infrastructure. So with this I will finish, and thanks for your kind attention.
Thank you very much, Dr. Parveen. I think that was an excellent and very comprehensive presentation. And obviously, we have a lot of questions also. So our first question is from Mr. Gore. He's talking about your first initial slides, like how many days it takes. You said that the virus attacks the olfactory nerves. So
yeah.
How much time it typically takes to get the senses back for smell and taste? Like, what is the average?
I don't know what is the time, but if I understand, if it is infecting first the upper respiratory tract, and it will stay with us up until the infection, until unless it is clears up. So I guess 2 weeks at least, if it is. But if it is reaches to the severe or moderate disease, then it will replicate in the lower respiratory tract, and then possibly even for the severe patient, after 2 weeks, it will go away. So I think possibly 2 weeks, but this is, I'm just speculating. I don't know exact number.
Okay. The next question is, what are the efficacy rates of our own vaccine, Covaxin and Covishield?
Yeah, that I think I listed. I can actually keep it on so that it's easier. Put the slide.
Sure.
Yeah. So for Bharat Biotech, we don't know, because for Bharat Biotech, the Covaxin, right? Yeah, Covaxin that has no phase 3 clinical trial data yet published. It is ongoing, so we don't know the efficacy. We know the AstraZeneca's efficacy, that is 80% if it is given above 8 weeks interval time.
So the next question is from Aishwarya Naik. Can you unmute yourself and ask the question?
Yeah, hello, ma'am.
Yeah, hello.
I actually wanted to ask 2 questions, but probably the second one you have answered. So my first question is, what factors determine the gap between the doses of the vaccine?
Yeah, so the thing is, this is again clinical trials, right? These are not fundamental studies. So it is very difficult to say in terms of fundamental studies, but I think this science paper where they are tracking people who got recovered, and you saw that possibly with a time of generally after 6 months the T cells are declining. So maybe if I can open the slide, you see there is a decline period. Yeah, so you see there is a decline period. So possibly somewhere in the half time one has to give, or something like that. How they are deciding after 8 weeks or 12 weeks, it is, I think, just from their intuition and from their knowledge, these clinical trials or earlier knowledge. But I think unless people do the cellular studies or this kind of animal model studies, then only they will know the reason behind what is working, you know. But possibly this is the reason. I think the immune system, how the T cells are declining over time, depending on that, you decide where will be the ideal to give it and give a boost.
And ma'am, as all the 3 strategies that we are now using, that those are non-replicative, as you said. So would it not be a good idea to use the adenovirus that is replicative instead?
Yeah.
So that we don't have to go for like vaccination in every.
Yeah, I understand what you mean. Yeah, but I don't know. because then you see with the viral vector is this also thing, although they are saying that people are not making antibody against the viral vector, but if it is start to replicate, we may produce antibody against the vector also. But you are right, possibly, because many vaccines we get, they are replicating, right? Or not, if not fully replicating, at least attenuated. That may also actually help. This is, I also think so.
Okay, the next question is from Oswald. You can unmute yourself and ask the question.
Thank you. So, is it not possible to innovate a medicine for COVID recovery rather than just managing with the immunization vaccine?
Yeah, that's what I was talking, right? That it is ideal that we should concentrate on designing a new class of treatment so that it can target the virus. For example, the antibody treatment, which is now, for example, these two antibodies, which is already in phase 2 clinical trials, and also this is already in emergency authorization. So the antibody-based drugs might be possibly more direct in neutralizing the virus, because other drugs, for example, the classical drugs, enzyme-based drugs, have not worked so far. So, but that I think we have to really do.
Okay. My next question is, ma'am, why do the virus mutate and when does this mutation stop?
Yeah, the virus, as I said, virus mutation is something it is not they have brain and they decide let's mutate. It is just a random process, and it's a evolutionary process, I think. And also, like from parent to progeny, they can have different mistake. Actually, they are actually kind of a mistake, you can say, and some actually somehow survive in the host better, and they start to persist. So it's a little bit evolutionary reason.
Oh, ma'am, and what about the Covishield properties? It seems that that regarding Covishield, it was not mentioned in your slide. Can you please give an overview of that?
The Covishield is actually the AstraZeneca vaccine.
Oh, okay.
In India, we are calling it Covishield, but it is, the name is this. This is the Covishield vaccine. Yeah. So yeah. So
Thank you,
this
ma'am.
is, yeah.
So the next question is from Akshat Mishra.
Ma'am, is there any vaccine made till now which can treat all kinds of variants?
Yeah, actually, that is interesting question. I was reading just recently. So now they are trying to make a spike protein nanoparticles. So, and then take different receptor binding domain of or some part of the spike of different variants and combine in one nanoparticle.
Okay,
so
ma'am.
that it will be like a kind of multi vaccine, multi-targeting vaccine. So I think that may come up sometime.
Okay, ma'am, would the technology you are talking of, would that be also applicable if any other variant will be found in the coming future?
Yeah, if for example, because they are kind of cutting and pasting them together, right? So if they can find out new variant and they can possibly synthesize it faster, but again, the clinical trials has to go on, and I really don't know.
Ma'am, any existing vaccine is not that much sufficient to cure all the viruses,
No, but
all the variants of the viruses.
No, but you see, this Pfizer is very good, right? This is what I was talking about. For example, yeah, wait, where is the slide? So this Pfizer, they really showed all major variants like UK, this is Brazil, and this is South Africa, slightly lower. Maybe they will now test for the Indian variant also. And Pfizer can really neutralize all three. Also, Bharat Biotech vaccine, Covaxin, and also Astra, but with slightly lower efficacy.
Okay, ma'am. Thank you, ma'am.
Next, we have Satyaki. Satyaki, go ahead.
Yes, ma'am. Ma'am, you told we may take vaccine every year as according to the T cell span and viral mutation. Then the vaccine has to rebuild every year. Is it possible? I mean, is it feasible actually?
Yeah, I don't know. That is like, but for flu shot, it is every year, right? So yeah, that but I don't know. I'm just saying that it may become like an influenza virus, and we may need to vaccinate at least for near future almost regularly.
Ma'am, one question
But
more.
this is I don't know. I'm just saying it. Okay, so there is no one has claimed this will happen or something like that.
Okay.
Because
Ma'am,
these clinical trials all are done very in short time, right? They have data only
yes.
for 6 months. So until unless we wait for 1 year, then we see the vaccinated people still have memory B cell, memory T cell, then we don't need. Then we will know, okay,
Yes.
we then don't need. So I think if we wait some time, then we will know it.
Yes, ma'am, one more question. Why post-corona diseases are happening? That is, after getting well, why a new problem arises for a corona patient?
Yeah, this long COVID, right? So
Yes.
yeah, I don't know, but yeah, this is difficult question. Maybe some of the viruses are not cleared as we are thinking. Maybe still they are there and then start to replicate again because we have not targeted them, right? So
Yes.
most of the drugs have targeted our immune system. So if we have not cleared the viruses, and if, for example, the patient have not made enough antibody against them, then we cannot clear all the virus. So they will be there with us for very long.
And ma'am, one more question. In the second mutant form that is happening in India, so that may sometimes give corona test negative, being positive. So why
No,
that?
no. Even if it is no, the PCR can detect even if it is mutated. So because it's only very some parts are getting mutated, not whole genome, and PCR is checking the whole genome. So it will be detected.
Okay, okay, thank you.
It is not a quasi species. It's not becoming a new virus. It is the same virus. Only few parts are changing. Very few.
Okay, ma'am. Thank you very much.
Next is Dr. Saptarishi.
Hello, Nagma. This is Saptarshi from ISA Bhopal.
Hi.
Hello. So thanks for the nice talk. Just a quick question. What percentage of the Indian population do you think needs to be vaccinated in order to attain herd immunity?
I don't know, but herd immunity is not related
The
with vaccine, right?
No, the reason I am asking is that after the second shot,
Huh.
most of the places in the US, they are saying that you don't need to wear the mask. So they are actually they're going the Spain or the Sweden model for the first wave when it started. So
Yeah.
do you think that by the end of this year we can also go mask free?
I
At
would
least when
like
say
to.
60 when 60% of the population is vaccinated. Yeah, I know it's a difficult call. Okay. Nice talk, Nagma. Thank you. Thank you.
I don't know, but I think it is better to be masked because you see these new variants are coming. And for example, even the Pfizer, who are so pushing towards their vaccine drive, they are vaccinated people having the antibody. They have lower neutralization for South African variant. So the one has to be maybe careful because we still don't know about these variants and whether they can transmit again, infect, and the vaccine may not work.
Right. Thank you.
So next is Deepak Bauman.
Yes, hello. I want to ask that how many days a virus can stay in our body?
So yeah, I think I showed one graph. So the virus replication cycle on average is about 15 days from the symptoms start. So I think this slide, yeah. So you see, right?
Yeah, 15
So
days.
if everything works, so if our immune system works, it will clear it within 15 days.
Okay. So if someone is positive with COVID-19, so after 21 days, we can assume that he or she is negative.
Yeah, should be.
Thank you.
Ideally, yes.
Thank
you. One question which I'm reading from chat box is why ivermectin is given, which is actually anti-parasitic, not like antiviral.
No idea actually. I also was thinking why they are doing. I think when in Italy and other countries and China, when they were seeing this huge rush of COVID patients, because these clinical trial data is on patients, right? Directly on the patients, on the hospital. The data come from directly from the hospital. So I don't know, means possibly the doctors have decided to give the ivermectin and combination drug. That's actually good. Why they come up with ivermectin? I actually don't know.
Right. So there are recent reports that steroid therapy, so you mentioned corticosteroid as one of the major drug used for treatment of COVID. And that has caused like a lot of these fungal problems like black fungus, white fungus.
Yeah.
Does it vary from like, are these fungal population ubiquitous in both indoor and outdoor environments, or like there are susceptible population only in certain geographical regions of the country?
No, but these fungus are everywhere. Like I think it's just sitting on the table. I don't know. They are very highly prevalent. But so it is everywhere around us. But I think this you see this in the PGIMR data, what they show that possibly when they are giving these steroids, dexamethasone particularly, then this immune suppression is very high, and this severe, moderate to severe patients are. I think Dr. Deepak?
Vishal.
Yeah, sorry, Vishal. Yeah, so Dr. Vishal also said, right? Also, we are having this giving the ventilation, and then the water. Is it the good water or is it distilled water? We don't know. And if it, as I said, it is everywhere. If it is in the water and if a patient is already immunocompromised, they are more susceptible. So how they are in the hospital treating patient, that also will introduce.
And this is a question from Dr. Ravindra. Can we kill virus in upper respiratory tract by any means?
Yeah, means you mean in the very early.
Yes,
yeah,
in the first few
days. Yeah, so if we have, that's why actually this I think that is why it actually worked. I'm guessing it, I don't know, this antibody treatment which was given for the mild patient. So maybe it has not reached to the lungs and it has not really started replicating in the lungs and only in the upper respiratory. So if it is given early on, such kind of antibody based drug or other drugs, antiviral drugs, which targets the virus, then possibly it can be cleared faster. So the problem with the SARS-CoV-2, the symptoms come only after 4 days of infection actually. So you don't know, maybe it already has passed on the lungs by then. So that is the problem. The symptom, if you compare for influenza, it comes within 2 days of infection. So it's much better to, easier to track influenza, whereas for CoV-2 it is salient for some time.
Okay, there is one question from Professor Vinod Singh. You can unmute yourself and ask the question. Professor Singh.
Yeah, Nagma, I just wanted to know whether alcoholic drink can help in the beginning.
I don't know, maybe. Yeah, why not? I don't know, but yeah, it is not in the standard care or therapy, so we cannot say.
Okay, thank you.
So one like kind of very simple question is, when you mentioned about cytochrome or storm or something, cytokinin storm.
Yeah.
So it's kind of we are suppressing the immune system to control that. But then once we treat the patient, like the immune system may remain suppressed for quite some time. So like we have fungal infection happening, the person is susceptible to other infections as well, right? Like
yes,
so it's
exactly.
like immunosuppressing. Any kind of disease can also find, like the patient may actually acquire. So you agree with that?
Yeah, so it is a little bit like people having diabetes type 1, right? Our body is acting against us. So it becomes a little bit like that it's so much we have a high immune like too much cytokine. That's why we have to treat the patient with anti-inflammation drugs. And then once it is too high dose or not adequate dose, then it can, other pathogens can take up and then cause other diseases.
Okay. So is there a data available, like how long it takes, like on average, people to recover and get the normal immune system?
If it is within 28 days recover, then it is going good. But if it is going downhill from 28 days, then it's going to just be severe. So that is kind of rule of thumb. So if it is within 20 days, if the patient is not getting better, that means it's just going to go downhill. Yeah.
Okay. Mr. Sanjay Chaudhary, you can ask questions. Please unmute yourself.
Hello.
Hello.
Ma'am, actually, I want to know how we can chemically inactivate the virus in vaccine.
In vaccine?
Yeah, there is a slide in which you can show the RNA,
yeah.
but it's chemically inactive.
I understood what you say. I understood. How it is inactivate, right? This propiolactone. Yeah. This propiolactone, that's actually a good question. I have to look into. So this propiolactone, if you look at the structure, it's this 4-membered ring structure, and it can penetrate the viral membrane. And as much I know that it actually somehow impairs the RNA of the virus, and how I don't know the mechanism now, but it impairs somehow. You can look, it is very well studied. It has been done for decades. So you can little bit look how propiolactone mechanism works.
Okay.
Yeah, so sorry, I don't exactly know the mechanism, but it works against the RNA. It kind of inactivates the RNA.
So with that, Dr. Nagma Parveen, thank you very much for giving such informative talk. And with that, we also come to the end of this COVID talk series. You can send to me or Professor Harish like comments and suggestions for the future. And I wish for everyone safe and healthy times ahead. Thank you very much and have a good evening. Bye.